Taxonomists of the Kerala Forest Research Institute have set out to study plants in their natural habitat. The expedition takes them into the hilly and mountainous tracts of the Western Ghats, which are endowed with a diverse ecosystem consisting of vast expanses of grasslands and prime forests of various types. Our taxonomists, however, are not looking for plants of the ordinary type. Neither are they out to investigate the floral diversity of the wild in all its resplendent beauty. They are looking for a unique kind of plant. These plants often go unnoticed as they lack roots and shoots common to the plant kingdom. These unique plants are called the lichens. Of the 18,000 to 20,000 lichen species which are known to lichenologists worldwide, the Indian subcontinent is reported to be home to about 2,000 species of lichens. The major known assemblage of lichens is in the Indian region of the North and Northwestern Himalayas, the Central Himalayas, the Northeastern Himalayas, the Central Indian region, the Southern Indian region covering the Western Ghats, and the Andaman and Nicobar Islands. The tiny state of Kerala is another major repository of lichens that is now being investigated by our team of scientists. There are two broad categories of lichens. The microlichens are those that grow as an inseparable dull and dry patch on trees and rocks. The other category of macrolichens, though seemingly dull, stand out on the substrata on which they grow with easily noticeable morphological features. Lichens grow on a variety of substrata of soil, rocks, and trees. Those that colonize the soil are called TERRICOLOUS. The generally inhospitable rocks, however, are home to many a lichen. SAXICOLOUS is the name given to lichens that grow on rock surfaces. The bark of trees, both small and big, serve as a good substratum for lichens. And those lichens that grow on the tree bark are called corticolous. Some lichen species grow on leaves. Such lichens are known as FOLIICOLOUS. Lichens also grow on a variety of marine life, like for instance the sea turtle. These dull-looking plant species, however, are a product of a complex symbiotic relationship between algae and fungi.
Lichens are a complex group of plants without vascular tissues. They do not have well-defined root, stem and leaves. Instead, they have a complex thallus arising out of a natural symbiosis between an alga and a fungus. Both of these could be seen through a microscope.
For instance, a cross-section of thallus of foliose lichens shows an upper cortex which is comparable to the epidermis of a leaf. Beneath it is the algal layer which produces food. In the center is the fungal layer called the medulla. Attached to the medulla at the bottom is the lower cortex.
Lichens are not considered as parasites, though they grow on trees. The food is produced by the algal component.
It is the fungus that protects the food-producing algae to constitute the thallus that is seen in its various forms on the substrata. Studying plant population in their natural habitat often involves a long and tiresome trek through the forest. Having reached deep into the forest, our team of lichenologists has decided to spend the night there and to continue the investigation the next day. Lichens do not grow in isolation, but in communities forming a large colony. They attach themselves to the substrata by hair-like bodies. Based on the extent of attachment to the substratum and the arrangement of tissues in them, the lichen thalli are grouped under 4 growth forms: CRUSTOSE, SQUAMULOS, FOLIOS and FRUTICOSE. In CRUSTOSE lichens, the whole of the thallus is attached close to the substratum and the thallus is extremely thin without distinct lobes. In the SQUAMULOS lichens, the thallus is an intermediate form between CRUSTOSE and FOLIOS forms. The thallus generally has minute lobules. In this species, for instance, the lobules of the primary thallus are white. The erect root-like bodies represent the secondary thallus. The folios or the leafy thallus of lichen is freely branched and extends outwards. The lobes that are irregular in outline have distinct upper and lower surface. Arising from the lower surface are minute bodies called rhizines, by which the thallus attaches itself to the substratum. Fruticose lichens are the most conspicuous, with the thallus branching out, giving it a shrubby appearance. In some species, the branches are cylindrical. In some, thread-like. Some others have a broader ribbon-shaped thallus.
Though not attractive, lichens are of economic value. Lichens are an important source of about 600 chemicals, some of which are used in the preparation of drugs, perfumery and dyes. Litmus that is used in the laboratories to test the acidity or alkalinity is derived from lichens.
Lichens grow very slowly. Their annual growth rate is just about 4 millimeters. This has helped scientists in estimating the age of the substratum.
In recent years, scientists have found lichens to be important agents in monitoring environmental pollution. It has been found that excessive emission of sulfur dioxide affects the photosynthesis in lichens, followed by their degeneration and death.
There is yet another abode of lichens on top of a mountain that our researchers seek to explore. Out there, it is foggy. But that is no deterrent to our team, as the spot is abundant in lichens belonging to various species. Here is a group of Heterodermia incana, a fruticose lichen. This golden yellow and freely branched lichen is Tilostschistus flavicans. This bushy one is called Usnea. Though hairy like the Usnea, this one belongs to the Ramalina rostleri species. And this patch of green lichens with irregular lobes is known as Parmelia.
These tiny lichens have different morphological structures. For instance, on the margin of lobes, there are hair-like outgrowths known as cilia, which are the organs of attachment. These small powdery balls on the tip of the lobes are called soridia. These are the propagules of vegetative reproduction. Here are cup-like structures. These are the fruiting bodies which contain spores for sexual reproduction.
The fruiting bodies do not look alike in all lichens. While in some they are like tiny flowers, in others they are cup-like. In yet others, they are button-shaped. Fruiting bodies in certain species appear as black dots on the thallus. Lichen species do not only live in colonies. Several species grow shoulder to shoulder with one another, covering an entire tree. An important part of studying plant populations in the wild is to note down all parameters of their habitat. In this case, it is the nature of the substratum, altitude, and other morphological features that are carefully noted. Samples of lichens for preservation in herbarium are collected along with the substratum. It could be a rock, as is the case here. If the sample to be collected is on a tree, the bark along with the sample is chipped. Sample collection is not the end of the study. Now the scene shifts to the laboratory, where the researchers set about identifying the collected samples for preservation in the herbarium. For this, the researchers examined the minute details of the samples. By using a key compiled by earlier researchers, each of the morphological features of every collected sample is compared. It is then classified as belonging to one or the other species.
Apothecia. This is Pseudocephalaria aurata, which is a new record to Kerala.
The KFRI researchers have identified 254 species of macrolichens under 43 genera, of which 10 species have been found for the first time in India. The samples so identified are kept in the herbarium, which are available for research purposes. In India, Lucknow has 3 herbaria. One at the National Botanical Research Institute, another is a collection of Professor D.D. Avasthis, and the third is at the Lucknow University. The other herbaria in India are at Kolkata and Shillong, and the latest in the list is the herbarium of the Kerala Forest Research Institute at Pichi. This then has been the story of the lichens. Next time you happen to see one, make sure you take a better look. They are worth looking at.